Hello everyone, my name is Kong Shrinani and welcome to my YouTube channel NeuroChat with AG. On this channel, I plan to cover topics that are closest to my heart and support the neurodiverse community. Topics related to neurodiversity, autism, late autism diagnosis, and autism in women and girls. I will interview thought leaders, esteemed researchers, scientists, professors, doctors, and psychologists and seek their views and updates on their latest work and research. I will also chat with parents of neurodiverse kids and adults and talk about experiences of neurodiverse individuals. Lastly, keeping inclusivity as a topmost priority, we might also discuss controversial disparate views for a collective understanding of issues and respect for diverse views. All the views and topics discussed are for informational purposes only. If you are interested in watching more quality content on this topic, please hit the subscribe button. Don't forget to press the bell icon to be notified whenever a new video is uploaded. Please also share the channel with your friends, family, and social media platforms. Thank you. Dr. David AMARAL is a prominent neuroscientist known for his significant contributions to understanding the neurobiology of autism. He is a distinguished professor at UC Davis, affiliated with the UC Davis Mind Institute, a leading center for research on neurodevelopmental disorders. Dr. Amaral has spent decades investigating the brain mechanisms underlying autism, focusing mainly on the role of brain structures such as the amygdala and other limbic system components involved in emotion regulation, social behavior, and memory. In addition to his work at UC Davis, Dr. Amaral is the founding director of Autism BrainNet, a program of the Simons Foundation that is a nationwide initiative designed to support the collection, storage, and distribution of post-mortem brain tissue from individuals with autism and other neurodevelopmental conditions. The project aims to provide essential resources for researchers worldwide who are studying the neurobiological underpinnings of autism and to improve the understanding of how genetic and environmental factors contribute to the development of the condition. Through his work with BrainNet, he has significantly advanced the field of autism research by facilitating access to critical biological samples, which are key to uncovering the neural mechanisms and potential therapeutic targets for autism. Dr. Amaral, welcome and thank you very much for the time today.
Nice to be with you, Akansh. Thank you for the invitation.
And to jump right into it, could you please talk a little bit about your background, what kind of started and got you involved in the field of autism, and what you've done ever since?
Okay. Well, thank you. I was trained as a neuroscientist. My PhD is in neuroscience. I also have a PhD in psychology, but I've always thought of myself more as a neurobiologist, neuroscientist. And most of my work for the early part of my career was trying to understand the brain regions that are involved in functions like memory, and emotion and social behavior. So the brain regions that are most involved in those kinds of functions are, for memory, the hippocampus or the hippocampal formation. It's a bunch of structures in the temporal lobe. And for emotion, it's another structure called the amygdala that you mentioned already. And so for years and years, I studied how those parts of the brain are connected with other parts of the brain and trying to come up with a story of how, for example, the hippocampus might make memories. And we did this by studying connections in animal models. But we also did studies of the human brain for people who had damage to one or the other structures and lost the ability to make memories. They had what's called amnesia or anterograde amnesia. So I did that for many, many years. And in the late 1990s, there was an upswelling of interest by parents in researching what was going on in autism. There was the sense that there were more and more people being diagnosed with autism. In fact, some parents thought there was sort of an epidemic of autism taking place, and there weren't enough answers about what was happening to cause this increase in the number of diagnosed autistic individuals. And there was relatively little information. People knew that autism was related to changes in how the brain functions, but there was relatively little information about exactly what was going on in the brain that was different in an autistic individual from one that was neurotypical. Um, there was a little bit of a hint that the amygdala might be important. And actually, uh, I got into autism initially because families contacted me and said, "Hey, Dr. Amaral, you work on the amygdala, and the amygdala is involved in autism, so you need to start researching autism." It was really being solicited by parents that I initially got into autism research. Initially, did a little bit of work trying to look at the brains of individuals that had autism during life and who had died and contributed their brain for research. But we really just started scratching the surface on trying to understand what differences there would be. And then in late 1990s, parents in Sacramento area, where UC Davis is located, decided they wanted to build an institute dedicated to autism research, and they called that the Mind Institute. And I was asked to be the founding research director of the Mind Institute. We really got going in 1998, and I have been working at the Mind Institute ever since. Continued to do anatomical studies, but also have been doing one of the largest projects that we have at the Mind Institute. It's called the Autism Phenome Project, where we're trying to understand different types of autism. And, I'm sure you and your listeners know that autism is very heterogeneous, and we're trying to understand that by doing comprehensive analyses of a large population of people with autism and trying to see what are the similarities and what are the differences that might actually make different meaningful subgroups. So we started that in 2006. I understand that you interviewed my colleague, Dr. Christine Nordahl, who started as a postdoc with me in 2006 and now actually runs the Autism Phenome Project. So she stayed with us almost into her entire career. I have also participated in attempts to create animal models of autism. Most recently, we're using genetic engineering techniques to produce animal models that have genetic mutations that are known in humans to often cause autism. That's another whole topic. Probably we could spend an hour talking about that. But those are some of the things that I've been doing. My career sort of swerved away from doing basic science on memory and emotion to really focusing on trying to understand what are the neurobiological underpinnings of autism, with the goal ultimately of trying to reduce reduce the disabilities that are associated with autism and improve the quality of life of autistic people.
Thank you. Yeah, that's really interesting, especially that last thing you mentioned. If we have time, I'd love to ask you more about that.
Sure.
So could you share a little bit more about the work that goes on within it and I guess how it's progressed from when it started to where you are now?
Yeah. There's a large number of faculty members doing different kinds of research studies at the Mind Institute at UC Davis. We started from scratch, so we were able to recruit people who were excellent clinicians, who were good diagnosticians of autism. We also recruited people who were more biologically oriented. And I think the two things that differentiated the Mind Institute from a lot of what was going on, particularly in the early 2000s, is that we wanted to study all forms of autism. So in particular, we wanted to focus on what is now called profound autism, individuals who have either low IQ, who may be nonverbal, When we first started the Mind Institute, those kind of individuals were generally excluded from research. So, since I was trained as a neuroanatomist, I wanted to understand the development of the autistic brain. Um, and the way you can do that in living individuals is carry out magnetic resonance imaging of the brain. And if you look at the studies that were done, even up until, the early 2000s, the vast majority of imaging studies that were done on autistic individuals were on very high functioning, very compliant individuals. Because, as you probably know, to do an MRI, you have to have somebody in a tube that is going to stay still for 10 minutes at a time in order to be able to get the imaging done. And most people thought, well, if you're somebody that had low IQ or if you didn't have language function, you wouldn't be able to do that. And we just didn't accept that. We decided that we needed to find ways to include those individuals. And so, again, with my colleague Christine Nordahl, beginning in the early 2000s, we came up with ways to allow children with autism, even if they had lower IQ or they had no language, to be successfully scanned for MRI. Initially, we did that by scanning the kids when they went to sleep at night. So we did virtually all of our scanning starting around 7 o'clock and sometimes going into the early morning when the kids would go asleep. And what we found was that if we were patient, we could actually be very successful. I think we're still around 90% success rate with kids who are 2 to 3 years old in getting excellent MRI scans. And then, of course, now we could do that with any child, whether they were higher functioning or lower functioning. And we've done that with all of our other assessments. So I think we were the first institute in the United States to really focus and find ways to carry out research on profoundly autistic individuals. So that was one thing. And I mentioned the Autism Phenome Project. We've now evaluated over 500 families in the Autism Phenome Project. And we have come up with different subtypes of autism based on different characteristics. And for example, I'm very interested in a group of kids mainly boys, that have autism and a brain that's larger than you would expect for their body size. We call it disproportionate megalencephaly. And interestingly, in neurotypical people, the bigger your brain, the more likely your IQ is to go up. So there's a positive association between IQ and brain size. But unfortunately, for these kids who have autism, the bigger the brain size, the more impaired they appear to be. So there's something going on there that we're still trying to understand, but we've been able to image a fairly substantial number of these kids with very large brains and autism that had never been really imaged before because they tend to be lower functioning. So that's the one aspect of the Mind Institute. The other aspect is that we tend to be very biologically oriented. So our parent founders were very interested in the idea that there may be types of autism that are caused by immune dysfunction. And so we hired immunologists who had not worked in autism before, but we said, try and investigate autistic individuals and see if you see immune dysfunction. And we, in fact, have found a number of things. For example, that a subset of people with autism, their mothers seem to have antibodies that are directed against fetal brain development. And that was a finding that came from the Mind Institute from Professor Judy Vanderwater, which was completely novel. And it was because the parents really were interested in us trying to look at biological causes of autism beyond what was already known. Um, Over the last decade, there has been a huge amount of effort also at treatment research at the Mind Institute. My colleague Sally Rogers created something called the Early Start Denver model, and she and her colleagues have used that for treatment of autistic individuals and have now transferred it to parent versions. So parents can actually be the intervention providers, and then to community-based programs where other providers in the community can use the same techniques. And our current director of the Mind Institute, Abin Stamer, is an expert in involving the community in treatment and advocacy kinds of efforts. So the Mind Institute is diverse. We do a lot of different things, and we study more than just autism. So that was the main focus, but there are other neurodevelopmental conditions like Fragile X syndrome. That there's been a quite large program, and others have been looking at Down syndrome and other genetic conditions that lead to neurodevelopmental disorders and are being studied as well. So, and it's, there are people housed at the Mind Institute. We actually, in the early days, built 2 buildings with 110,000 square feet of space to house the faculty and the clinics of the Mind Institute. But there are many people outside of the buildings of the Mind Institute at the UC Davis campus that collaborate with us on various kinds of projects, all directed at understanding neurodevelopmental disorders.
What are some of the key differences in terms of brain structure for people diagnosed with autism? Are there any common similarities that people who don't have autism don't exhibit? Because I know, as you mentioned, it's a very heterogeneous condition, but are there any similarities you've noticed across the board or for a large proportion?
Yeah, that's a really good question. And so far, I would say that, we see, so first of all, autism is a developmental condition, a developmental disorder, and things change over time. So if one of the problems with a lot of early autism research was that people would only be looked at one point, one time of their life, like a snapshot of their life. And really, autism may be a dysregulation of the normal development of the brain or normal development of behavior. And so that's why we thought it was so important to do longitudinal studies, in the Autism Phenome Project, for example. So, and there are different subtypes, as we've already talked about. So the big brain form of autism, for example, that we've probably done the most work on, for those kids, their brain actually starts showing enlargement above what you'd expect. And when I say above what you'd expect, it's based on normative curves that are taken from neurotypical. So you can think of like a growth curve in body development that, children go through when they go to the pediatrician office and they say, well, you're at 50% or you're at 95% or whatever. That's based on looking at loads and loads and loads of typically developing kids and charting how fast they say how tall they are and how much they weigh over time. Well, you can sort of plot on those charts where autistic kids lie. And so the kids with the big brains, what we see is even by 6 months of life, their brain is already bigger than you would expect. And what's interesting about that is that you actually don't start seeing symptoms of autism until around a year of life, or maybe even later, 18 months to 24 months. And you really can't, fully diagnose a child until around 36 months. So what for that particular form of autism, what it shows is that the major difference between typical and autistic is that the brain's growing too fast. Now, what causes the brain to grow too fast? That's a question that we haven't answered yet. We don't know whether it's because there's too many neurons, which are the main, functioning cell type in the brain, or or too many glial cells, which are like supporting cells of the brain, whether there's been a failure of what's called the pruning process, because early on your brain grows too many connections and then some of them get cut back. We call it the pruning process. And it's normal, and it's like you want to make sure that the brain makes the right connections. So you can start off by making too many connections, and those that look like they're going to be functional stay, and the ones that are dysfunctional get eliminated. Well, maybe that process doesn't work out as well in an autistic brain for whatever reason, and that's why the brain is too large. We're still trying to figure that out. In terms of, but going back to your question, which is a really good one, can I point to one thing that's different in all autistic brains from neurotypical brains? And I can't. We haven't come up with that critical notion of, what really differentiates the autistic brain from the neurotypical brain. And I think in part it's because there's a lot of heterogeneity in typical brains, and there's a lot of heterogeneity in autistic brains. And we still are in the process, we haven't really studied enough brains in general to be able to figure that out. Maybe that'll come out as more and more and more and more brains are analyzed. But, we're still sort of in the early stages of trying to carry out the neurology of autism. So we have a bit ways to go yet.
Yeah. Okay. Thank you for explaining that.
Yeah,
And could you also talk a little bit more about Autism BrainNet and how that all started and how it's going now?
sure. Okay, so this is a little bit of a difficult topic. Um, how do you understand what's going on in the autistic brain compared to a neurotypical brain? And we've been talking about that a little bit. And when I talk about MRI, I sort of think about it as like looking down at the earth when you're flying. You're at 30,000 feet and you're looking down at the earth. You can see big patterns. You can see mountains. You can maybe see, cities and whatever, but you can't see the detail. You don't see individual people or cars or whatever. And we, in neuroscience, the MRI gives you that 30,000 foot view, but really we think that what differentiates the brains in autism from neurotypical are going to actually be seen at the cellular and molecular level. And so how do you get to the cellular and molecular level? Well, really the only way is to study the human brain of a person that had autism during life, and when died has donated that brain to something like Autism BrainNet, where we can then distribute that tissue to investigators to study the molecular biology and the histology or neuropathology of the brain. So why did we start Autism BrainNet? Well, for some neurological diseases like Alzheimer's disease, that has been studied for, well, actually, the first definition of Alzheimer's disease took place more than 100 years ago by Alois Alzheimer, who was a neurologist and psychiatrist, and he came up with the idea that there were profiles in the Alzheimer's brain that really differentiated it from the brain that didn't have Alzheimer's disease. These are called plaques and tangles. But since then, literally thousands of brains have been studied from people who had Alzheimer's disease, and when they died, they contributed their brains. And we know a tremendous amount about what leads to Alzheimer's disease. And in fact, most of the treatments now that are being tried for Alzheimer's disease or the preventive measures that are being worked on as preventatives for Alzheimer's disease has all come from post-mortem brain research. So now if we talk about autism, it turns out that that analysis of the autistic brain really only started in the 1980s. So, about 40 years ago. And the number of brains that have been looked at, a very, very small number. And in fact, I mentioned when we first started that I started in autism research by trying to look at the autistic brain, and really there was like 10 brains available to be able to do studies, which are not enough, particularly given the heterogeneity of autism. And I used to go to a meeting called the International Meeting for Autism Research, and every year we'd have like a think tank about doing postmortem research. And instead of talking about all the new findings that were coming out, everybody just kept talking about the fact that there weren't enough brains to study, and everybody was frustrated about that. So one thing led to another, and we decided we have to do something to gather more postmortem brains for researchers to do state-of-the-art research. And so I went to the Simons Foundation, which is a large private foundation that funds autism research. It's based in New York City. And I went to Autism Speaks and tried to convince them that they should invest in developing an enterprise to advertise to the community that it was necessary to get postmortem brains and to set up the infrastructure if somebody wanted to donate a postmortem brain, to bring in the brain, to preserve it properly, and then to make sure that we safely stored it so that we could then send tissue out to different investigators around the world. And, again, fast forward after lots of discussions and whatever, in 2014, we launched Autism BrainNet. We have collection sites in 3 places in the United States. One of them is at the UC Davis Mind Institute. One is at UT Southwestern, University of Texas Southwestern in Dallas. And the last one is now at Mass General Hospital in Boston. We also have one Canadian collection site at McGill University. And so the bottom line now is that we spend a lot of time, and one of the reasons that I like to do, interviews with people like yourself, is because I'm trying to bring the message to the autism community that postmortem research is really the way that we're ultimately going to understand what causes autism and could lead us to coming up with more effective targeted treatments for some of the disabilities of autism. And I'm not saying cure autism. What I'm saying is that, we would like to understand why so many people with autism have anxiety disorders. And can we come up with a strategy for helping those people to reduce their anxiety, or sleep problems, or epilepsy, all of which is associated with autism. So our belief is that by studying the postmortem brain, we will get to those answers that will help people with autism. And since 2014, we've now collected more than 300 brains. And we have started distributing tissue from those brains all over the world. In fact, as far as Australia, into Europe, to many investigators in the United States. And now new research is coming out, using that tissue and demonstrating some of the facts about autism. We get good, it's difficult because, of course, if we're getting a brain tissue, that's because somebody has died. And it's obviously it's a tragedy for the family. But the families often tell us that by making a donation of their loved one's brain, that they feel like something positive has come from what would otherwise be a really tragic situation. And so on our website, which is autismbrainnet.org, you can go and under the Families tab, you can see that there's a Memories of Hope section. And many of our families talk about the person who donated their brain. And when we communicate with them, they say, we're happy that we have been able to make this contribution because we know that it will help autistic people in the future.
If people wanted to say donate their brains or the brain of a family member at their time of death with consent, how could they do that?
Thank you for asking. That's a good question. So we have a 24/7 hotline. The number is 877-333-0999. Just say it again, but maybe you can post it too. It's 877-333-0999. And a family member or a family physician can call that number anytime, day or night, and they will be put in contact with one of our clinicians. And the clinician will talk them through the entire process. So, as a first step, the family has to consent to making the donation. And then once that occurs, once the consent is acquired, then the clinician will handle every aspect of the donation. So the clinician will find somebody in the area where the donor is located to acquire the brain, to ship the brain to one of our collection sites, and then the clinician will contact the family afterwards, whenever the family is available, to ask questions about the donor in terms of confirming their diagnosis of autism, but also getting more information about them. Did they have intellectual disability? Did they have, other issues that we should know about? All of which are important for researchers who are, who are going to be carrying out the research. It's important for anybody to know, and there's what we tell people is that this is important for autism research, and we would like either autistic individuals or family members of autistic individuals to talk about the idea of donation ahead of time, because this is not something that you want to start discussing at the time of a death. We know that that's a very emotional time. There should be a decision made before that happens. And then if you're an autistic person and you want to make a brain donation, you should tell your next of kin that that's your wishes, and so that they can implement that. Brain donation is different from organ donation. So if, for example, you indicate on your driver's license that you want to be an organ donor, that's terrific. And organs that are donated go to living people, and they are to preserve life. If you're going to, donate a kidney or a liver or a heart, that goes into somebody else in order to allow them to continue living. Obviously, we're not doing brain transplants, and so brain donation is only for research. And if you already say that you want to be an organ donor, that doesn't mean that your brain would also be donated at the same time. You have to, in addition, say that you would like to donate your brain to research. And so, again, once you've made that decision, then by calling the 877-333-0999 number, which is on our website, we will make sure that everything happens. There's absolutely no cost to the family. There's no religious prohibition of being a brain donor. Some people ask that. Some people ask, well, can I have a funeral like normal, like an open casket funeral? And the answer is yes. That doesn't preclude any of those things. So we try and make it as simple and as least anxiety provoking as possible to be able to make a brain donation. And then once that donation comes into Autism BrainNet, it's important for people to know that it supports research uh, not in a single study, but in dozens, if not 100 studies or more. And the tissue is available for decades to be used for research into autism. So it's a very, very valuable contribution.
Regarding, I guess the value of the contribution, another thing I wanted to ask is, once you have, the brains and like, you're doing research on them, what kind of specific discoveries have, these brain samples made possible?
There's loads and loads of discoveries that are being made now based on postmortem research of the autistic brain. But I'll just give you or I'll give you a couple of examples. One is related to genetics. So it's known now that there are, maybe as many as 200 genes that if you have a mutation of one of those genes, you're going to be more likely to have an autism diagnosis. This is all generally based on looking at the genes from either blood or other samples that are not related to the brain. And that has been, that's been enormously useful research. But what has been shown in the last 10 years or so is that if you start looking at the DNA within the brain, you can actually have local mutations of genes that you may not see based on blood samples. And the reason for that, it's called somatic mutations. And what that means is that when the neurons are dividing early on, and there's a period of development when the human brain is producing 25,000 neurons a minute. So, it's like enormous numbers of neurons turning over and dividing. And what happens when you have that kind of activity is every once in a while there's an error. The DNA doesn't get copied properly. And you can have a local mutation, and that mutation gets recapitulated in successive divisions for a while, and that may only be in a small number of neurons located in one part of the brain. And these somatic mutations, turns out they're more numerous in the autistic brain. And it may well be, and this is early stage of this kind of research, but it may well be that these somatic mutations, which are not reflected in blood samples, are part of what's causing the behavioral changes that are associated with autism. So we need to, there's a lot of work that's going to go on between finding these somatic mutations and drawing a real causal link to autism, but that's something that wouldn't have been known without studying the postmortem brain. That's not going to come out of any animal studies or MRI or anything else. You have to look at the autistic brain. Another example is that based on not genetics, but actually looking neuropathologically at the cells in the brain, is that basically there's two kinds of cells. There's excitatory cells and there's inhibitory cells. The excitatory cells are the majority of cells in the brain, like all the pyramidal cells in the cerebral cortex are excitatory. And then there's a second class of cells that are called inhibitory interneurons. And they form circuits with each other, and they produce a balance between excitation and inhibition. And that balance is necessary for normal functioning of the brain. And so there's been a theory going on for some time now that there's a perturbation in the excitatory and inhibitory balance, and that might actually lead to some of the behavioral alterations of autism. And in fact, as you probably know that 20 to 30% of autistic individuals also have seizure disorders, and seizure disorders are also due to a reduction in the inhibitory mechanisms. So the excitatory activations are sort of go unchecked and cause the seizures, which are just really rapid and coherent firing of excitatory neurons. Well, it turns out, based on postmortem assessment of the number of inhibitory neurons in the brain, that it appears that there's a decrease in the number of, at least there's multiple inhibitory neuron types, but one of the major ones is called the chandelier cell, and there's fewer chandelier cells in the cerebral cortex of autistic individuals. This is studies coming from one laboratory, needs to be replicated. But again, you would never see this if you were to study the brains of animal models or in any other way other than looking at the brains of autistic individuals. So again, both at the cellular level, the histology, there's examples of differences in autism versus neurotypicals. And at the molecular level, at the genetic level, there's also differences that are only being shown by studying the autistic brain and control brains, brains of neurotypical individuals that are used as a comparison.
And then another question I had, diving more into the brain structure and research side is what have been like some of the surprising findings in your research, especially related to the amygdala and the limbic system?
There was early suspicion that the amygdala might be really important for social behavior and for some of the social disturbances that are seen in autism. And for years, we carried out studies using animal models, primarily the non-human primate. And what we were able to show is that we could disconnect the amygdala in the monkey, even in very young monkeys, and we didn't alter their social behavior. They could do normal social monkey social behavior, which is pretty complicated, even though they didn't have a functioning amygdala. But what we found was that their emotional responses were completely altered. So a monkey without an amygdala doesn't show normal fear. So even monkeys interacting with other monkeys, monkeys can convey their intentions by facial expressions. And so if one monkey threatens another monkey by baring its teeth and making an aggressive face, and if the other monkey is subordinate, the monkey should show fear and either run away or have what's called a fear grimace and show the dominant monkey that it knows it's subordinate and everything is okay. But our monkeys without an amygdala, they didn't get it. So if a monkey was aggressive, they didn't realize that they should be fearful. And in fact, if we didn't prevent it, they could get in trouble because the dominant monkey would say, what's going on? Why doesn't the monkey realize that they should be scared of me? So, and we would show over and over again that the monkeys without an amygdala were not able to process the emotion of fear properly. So how does that relate to autism? Well, we found in our Autism Phenome Project that as many as 70% of people with autism have anxiety disorders. And the field of neuroscience really has come to the conclusion that the amygdala contributes to anxiety disorders, and probably because it really is not appreciating, it's sort of hypersensitive to the environment. And things that shouldn't produce fear or anxiety are now producing fear and anxiety, and that's what an anxiety disorder is. And so when we looked at the amygdala of people of autistic people, what we found is that early on, the amygdala has too many neurons, potentially being hyperactive, and that later on, in adulthood, the amygdala actually has fewer neurons. And what we think is that the amygdala early on sort of is hyperactive, producing sort of an anxious state in autistic individuals. Uh, that unfortunately has a deleterious effect on the structure of the amygdala, and it starts actually degenerating to a certain extent. And so the person continues to have anxiety because it's sort of now learned that this is the way it should act. But the amygdala goes through this developmental process that is different than what happens in neurotypicals. So again, this is something that we hope is going to be replicated in other studies, but it brought us to the conclusion that the amygdala is involved in autism, certainly, but it's not involved in the social aspects of autism, but it's definitely involved in the anxiety component of autism. And again, if we understood it better, we may be able to come up with better interventions to reduce the anxiety in autistic individuals.
And
I want to say one other thing to Akash, that there are surprises, and one of the surprises that came out based on our MRI studies is that there seems to be a dysregulation of how cerebral spinal fluid is handled in the brain. So cerebral spinal fluid in the brain, there's these ventricles like canals that produce cerebral spinal fluid. It starts in the ventricles and then percolates out on top of the brain, and then eventually it gets out of the brain, out of the skull itself. It used to be thought that it was just a shock absorber to prevent the brain from getting damaged in the skull. But now we know that this cerebral spinal fluid really handles all kinds of substances that are necessary for normal brain functioning or normal brain development. And that actually removes toxins from the brain during life. And even to the extent that the cerebral spinal fluid seems to take toxins out of the brain more when you're sleeping. And one of if you don't sleep very well, it may impair the brain's ability to rid itself of some of these toxic substances. So we found in very young children who before even they were diagnosed with autism, but who are the siblings of autistic individuals and therefore at higher risk of ultimately having a diagnosis of autism, that they had increased cerebral spinal fluid between the brain and the skull, and that this was actually diagnostic of whether somebody was going to go on to have autism or not. This increased, we call it increased extraaxial fluid, has been replicated now a couple of times, and this was a complete surprise. We weren't expecting to see this. And it's something now in the field that people are paying attention to. It's not something that we need to look at necessarily at the postmortem level, but there are going to be things going on in the brain, particularly early on, that lead to this abnormal flow of cerebrospinal fluid, which may be contributing to other brain changes that ultimately lead to autism. So, you were sort of saying surprising. That was probably one of the most surprising things to me. We found that initially in 2013, and there was a graduate student in the lab, Mark Shen, who's now a faculty member at UNC, and he's continuing to pursue that to see what might be causing this increased extraaxial fluid and how could it contribute to the diagnosis of autism.
Okay, I think first I want to ask the thing you mentioned earlier in regards to animal research with adding those altering those genes with genetic engineering, and that you might see in people with some people with autism. So if you could talk a little bit about that.
Sure. So let me first say that I believe animal research is necessary. There are things that ethically you wouldn't be able to do with human patients to understand conditions and diseases, and so animal research to really get us to the point of understanding the biology of conditions like autism or cancer or whatever that you might be interested in. We do need to do animal models. On the other hand, I'm very respectful of animals. And I think, we definitely need to use the appropriate animal model, and we need to use the fewest number of animals possible in order to get the results to answer our questions. So I mentioned the big brain form of autism before, that I'm particularly interested in this because it causes such a profound form of autism with often low language, lower IQ, and very little progress over time. And it turns out that it's not genetically homogeneous. It's not only one genetic mutation that causes the big brain form of autism, but there is one gene. It's called CHD8, that if you have a mutation of that gene, it's almost 90% likely that you'll have a diagnosis of autism, which is enormous. It's very penetrant. And if you have a diagnosis of autism, it's highly likely that you'll have the big brain form of autism. So, and this is all known from human clinical studies, some of which we've participated in. Now, the question is, what causes the brain to be so big? And I told you earlier that, you can look at the people that have this form of autism with even CHD8 mutations with the MRI, but it really doesn't get you to the cellular and molecular levels. So the only way you can really understand the biology of the big brain and CHD8 form of autism is to create animal models. And so many, many mouse models were created. But the bottom line is that none of them really mapped onto human autism very well. So, and it's complicated why that is, but in many respects, the mouse is not the best model for human autism. Its brain is so different, its biology is so different that it really isn't the best animal model. So I mentioned that we had been doing quite a bit of work with monkeys, with rhesus monkeys, and I decided that for many reasons. So the rhesus monkey's brain is about a tenth the size of a human brain, but it basically has all the parts. So there aren't any parts of the human brain that we don't see in the monkey brain. That's not true for the mouse. And the monkey has a complex social behavior. It uses things like facial gestures and other ways of communicating social intention. So I thought, well, this really is the best model for trying to understand the biology of human autism. Now, how would you make a monkey model? Well, there are different strategies, but I came to the conclusion that in this case, the best way to do it is to create a mutation that we know causes autism in humans. And the mutation that's most likely to cause autism in humans is a mutation of CHD8. And so my colleagues and I, for the last couple of years, have been working on using CRISPR technology. CRISPR is a way to introduce a mutation into the genome of a monkey. And what you do is basically you construct a vehicle that you can inject into a fertilized egg. So this is now taking it all the way back. You have an egg and you have a sperm. You can fertilize the egg, and then you can inject the CRISPR reagent into the egg. And the CRISPR reagent will go and snip out a little piece of the DNA that you've designed it to do. And so in the case, we are snipping out a little bit of CHD8. And when you snip out that little bit of CHD8, it becomes non-functional. And so and of course, you get two copies of CHD8. We knew from the human studies that if you damage both CHD8 alleles, they're called, then the embryo doesn't live, that it's too damaging to the development of the rest of the embryo and fetus. So we've designed it so that we're only damaging one of the CHD8 alleles, the other one stays intact. And then we do in vitro fertilization, so it goes through in vitro fertilization initially. And then just like in human IVF, we grow up the embryo until it's a certain stage. We then freeze it down until we have a female that's receptive, and the embryo is implanted into the female. And if we're lucky, the embryo implants and produces a fetus. And if we're really lucky, then the fetus would go to term and we would have an infant that would have that mutation. And at the upcoming International Society for Autism Research meeting that takes place in April, I'll be announcing our results of that line of research. I can't tell you what happened here, but we're enthusiastic about that. And of course, then what you can do is all kinds of biological studies with the monkey models, understand more about what's going on in the brain. Are there more neurons? Are there more glial cells? The kinds of questions that I mentioned earlier. For the first time, we'll be able to answer those questions using that animal model. We're doing other work too with animal models where there's another condition called Angelman syndrome. Angelman syndrome, we know the genetics that causes that. And it's sort of an interesting issue because in Angelman syndrome, the condition is caused by the fact that normally there's a gene called UBE3A. And that gene, of course, you get a mom UBE3A and a dad. The dad UBE3A is silenced, so it doesn't function. That's the way the normal situation is. And it's silenced because we call it epigenetic silencing. Something blocks it from functioning. So only the maternal UBE3A gene works. But if something goes wrong and the maternal UBE3A is mutated, so it can't function, then you can't produce any UBE3A protein. So people have already figured out how you can correct that. And it turns out if you can unblock the paternal gene, then it starts producing protein. And it turns out that if you're a mouse and you do that, you're okay. It doesn't produce the Angelman syndrome. Now in humans, normal kids who have Angelman syndrome, they have a small brain, they have severe epilepsy, and they just don't do well. They actually die early. But there's the notion that if you could do that gene therapy with kids who were diagnosed prenatally with Angelman syndrome, you could actually prevent them from having the disease. Now the point is, though, people don't want to go from mouse models to human genetic engineering because it's too big of a leap. So with colleagues from University of North Carolina, we're working up at a couple of primate centers to produce an Angelman syndrome monkey with the goal of trying some of these prenatal genetic therapies and show that they're both safe and effective. And if that works out as we think it will, then that would lead to more likelihood that it would be able to be tried in human kids who have this, who are going to have Angelman syndrome, but if we can treat them, not. And I'm really, really excited about that component of this whole program because it means that we may actually be within the not too distant future, having the ability to prevent a very serious, debilitating, and life-threatening disease from occurring in at least a small population of children.
Like looking forward, what are kind of your, I guess, goals or hopes for the future of autism research?
Yeah, there's lots of different goals, and when we when we first started the Mind Institute, I knew almost nothing about autism, and so when the parents told me they wanted us to cure autism, I thought, okay, we'll we'll work on it and try and come up with a cure of autism. And that was in the 1990s. Since then, I realized, particularly, being educated by many autistic individuals themselves, that they don't necessarily want to be cured, but they want some of their problems to be reduced, just like everybody else does. And so, you know, I'm I think number one, I hope that my you know that autism will be we're coming up in April to you know Autism Recognition Month, and I hope that people will um, acknowledge that autism is a human condition, that there are both positives and negatives related to autism, and we should accept autistic individuals in our culture, in our community. I do think that we need to understand the biology of autism, particularly autism that affects profoundly autistic individuals, because they have so many disabilities that I think it's right to say that most parents of a profoundly autistic individual would like a lot of their disabilities to be cured, to be so effectively treated, that they no longer bother that individual. And I'm talking about things like, intellectual disability. I'm talking about things like profound, like prominent anxiety, epilepsy, sleep disorders. So I'm hoping that the work that we do at the Mind Institute and throughout all the autism research centers, not only in the United States but around the world, leads to decreased disability in autistic individuals. And I think we'll get there, but I think what I've realized is that because autism is so incredibly complicated, it's going to take a while. And it's in many areas we're really just beginning to scratch the surface and understanding what's going on in autism. And so I think it's going to take many generations of autism researchers. But it's it I what I've found and what I'm encouraged by is that people who study autism are passionate about wanting to help and wanting to support the autistic community. And I'm certain that the people at the Mind Institute that I work with, that's their primary motivation. They want to make things better for the autism community.
My last question would be, is there anything else you'd like to share?
Well, the only thing we didn't talk totally about Autism BrainNet, but I would like to encourage people who are got this far in this in this conversation to go to our website. So it's autismbrainnet.org and learn a little bit more about our organization and what we do. See the videos of the families that have contributed to Autism BrainNet. Understand a little bit more about the value of postmortem brain donation, and then if you're an autistic individual yourself, make a decision about whether you think it's a good idea to do postmortem brain donation. If you're, the family of somebody who has autism, have a discussion about it. Think about whether it's a good thing to do. And if you do, contact Autism BrainNet. We have a newsletter that comes out about quarterly You can sign up for it. There's no obligation. There's never an obligation, and you that we don't have a pre-registration at this point in time because we didn't want to make people feel like if I sign up, am I obligated to do this? What if I change my mind? There's never an obligation. People can change their mind all the way to the last minute. But if anybody has a question about brain donation, they can contact me directly, or there's an email. It's called info@autismbrainnet.org. That's info@autismbrainnet.org. Put a question there and we'll get back with an answer as soon as possible. So thank you so much for your questions. I really enjoyed talking with you.
Yeah, thank you so much for taking the time to, share all of them and your expertise on all these topics. I really did learn a lot in this interview about like a lot of things related to autism research that I didn't know about.
Terrific, terrific. So nice to be with you today. Thanks so much.
Thank you.